Hello everyone and welcome to my Advanced Concepts presentation for Module 4. I am Miranda Pickett and let's get started. So today for the Advanced Concepts presentation for Module 4 we're going to be talking about the synthesis of acetyl-CoA from pyruvate. And I've put this image here. I thought it was pretty much the best image I could find to just kind of give a generic overview of what's going to be happening with the pyruvate dehydrogenase complex. So what we're going to be talking about today initially is the background information. I think there's a couple of things that are just good to know before we start getting into depth with things. I'm also going to be talking a little more in depth about the enzymes and coenzymes. There's also two preparatory steps that have to be concluded before the actual complex processes can start. And then I'll give a brief overview of the three main steps, and then we'll get into the steps. Moving right along, I'm going to move my camera here, probably better over here. So cellular respiration consists of three main processes, and these include glycolysis, the citric acid cycle, and the electron transport chain. The process of creating acetyl-CoA from pyruvate is what forms the connection between the first process, which is glycolysis, to the second process, which is the citric acid cycle. The main player in the conversion of pyruvate to acetyl-CoA is the pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex has three distinct enzymes, and each of those enzymes all have their own very own active site. The structure of the complex and its enzymes allows for easy flow of the substrates so that they can travel in an efficient manner from one active site to the next, making the process very efficient and quick. And lastly, the pyruvate dehydrogenase complex also has 5 coenzymes, and we're going to go over all of those in the next slides. So I first wanted to go over the coenzymes. The coenzymes are thiamine pyrophosphate, or TPP, lipoic acid, or lipoamide, flavin adenine dinucleotide, and coenzyme A, which can also be called CoA, and then your nicotinamide adenine dinucleotide. It is 3 o'clock in the morning. I'm so sorry. So for TPP, it is catalytic, which means that it's not permanently altered whenever it's used in the reaction, and it's used in the enzyme pyruvate dehydrogenase, which is E1. Lipoic acid, or lipoamide, is catalytic as well. It's not permanently altered from the reaction, and it's used in the dihydrophil transacetylase. Flavin adenine dinucleotide, or FAD, is also catalytic, and it is also not permanently altered in the reaction, and it's used in the dihydrophil dehydrogenase. Coenzyme A, or CoA, is stoichiometric, and it serves as a substrate. So it is permanently altered from the reaction, and it's used in the dihydrophil transacetylase. I really can't talk right now, can I? Dihydrophil transacetylase. And lastly, we have the nicotinamide adenine dinucleotide. It also serves as a substrate, and it's used in the enzyme dihydrophil dehydrogenase. Moving on to our three main enzymes. The first one is pyruvate dehydrogenase. It's the E1 enzyme in the complex. Its prosthetic group is TPP, and the reaction that it catalyzes is the oxidative decarboxylation of pyruvate. The second enzyme in the complex is dihydrophil transacetylase, and its abbreviation is E2 with a prosthetic group of lipoamide, and it catalyzes the reaction of the transfer of acetyl-CoA to coenzyme A, or CoA. The last third and last enzyme in the complex is dihydrophil dehydrogenase. Its abbreviation is E3 with a prosthetic group of FAD, and it catalyzes the reaction of regeneration of the oxidized form of lipoamide. So there's two preparatory steps that have to take place before the pyruvate can actually make it to the mitochondria and start with the process. The first one is that the pyruvate has to be generated through glycolysis pathway, and this, as we know, takes place in the cytosol of the cell, cytoplasm of the cell. The second preparatory step is that pyruvate has to be transported from the cytoplasm into the mitochondrial matrix, and it has to be here in order to be converted into the acetyl-CoA. So an overview, there's three basic steps that we have going on here, and the very first one is the decarboxylation, the second one is oxidation, and third is the transfer of the CoA. So to get into more depth, I have a picture of step one here. I'm pretty sure that it's the same picture that's in our books, but I did have to find it online because I no longer have the electronic book. So as you can see, my references are beneath on the pictures. But for step one, as I said earlier, it involves decarboxylation. It's basically that pyruvate will combine with a carbon ion of TPP, and then it goes through the decarboxylation and it has a product of hydroxyethyl TPP. And it's important to note that step one is the regulatory step of the process. For step two, as I said earlier, it's oxidation. So from our product of step one, which was our hydroxyethyl TPP, the hydroxyethyl group will be oxidized and will form an acetyl group, and then it's also transferred to the lipoamide. This creates an energy-rich thioester bond, and finally, the last part of step two is that the disulfide group of the lipoamide is reduced to form acetyl lipoamide. For step three, seen in this picture right here, this is where we actually get into the formation of acetyl-CoA. Going back from step two, the acetyl group is transferred from the acetyl lipoamide to form acetyl-CoA. And you can see that here. Excuse me. Those are the three main steps. Steps 4 and 5 are basically the steps that are resetting the cycle so that it can start again. So as I said, the purpose is to restart the cycle of acetyl-CoA formation, and they do this by oxidizing lipoamide, and they also have to reduce FADH2 back into FAD as well, and this is done through reduction of NAD+. So in conclusion, we went over a little bit of background information regarding the pyruvate dehydrogenase complex. We also went over the enzymes and coenzymes. We have 3 enzymes and 5 coenzymes. We also went over the 2 preparatory steps because we know that we have to get pyruvate by going through the process, the pathway of glycolysis in the cytosol before we can move into the mitochondrial matrix to begin the formation of acetyl-CoA. And we then went over just a basic overview of the main 3 steps in the complex. And then lastly, we went a little bit more in depth of the steps. My references are here on the last slide. I pretty much just used the textbook and got pictures offline. Thank you for listening, and I hope everyone has a wonderful night.